In January 2015, experts in cancer research from around the world gathered at the Riken Yokohama campus in Japan. They were there to take part in a special meeting organized by the EC-funded MODHEP project, probably one of the most comprehensive research efforts ever undertaken to describe the mechanisms behind liver cancer, particularly hepatocellular carcinoma, or HCC, one of the most common malignancies worldwide, and in essence, still an incurable disease. Riken plays a pivotal role in the project, bringing to the table its expertise in the computational analysis known as cage technology, needed to understand the complex datasets being produced during experimentation by the labs of its MODHEP partners based throughout Europe. The 5-day meeting in Japan had 3 main objectives: to offer special training to up-and-coming young scientists involved in cancer research of the cage technology devised at Riken, to provide a platform for discussing our understanding of liver cancer development at every level at a global symposium, and to host the 4th annual MODHEP project meeting, during which partners were able to discuss the progress being made, the implications of their work in relation to the overall aims, and to plan for the next stages for the remaining year and a half.
Is an in-depth, informal scientific discussion with critical insight of where we are and where we go from today till the end of the project.
MODHEP is an EU project funded through FP7. It is made up of 17 partner organisations from 8 different countries, 5 EU member states, Israel, Australia and Japan. The €12 million project started in 2011 and will last 5.5 years. The MODHEP consortium brings together world-class scientists and researchers with wide-ranging expertise, ensuring it has the skills required to achieve its ambitious aims and objectives. These are to provide genome-wide understanding of alterations occurring during initiation and progression of liver cancer, analysis of the genetic, epigenetic, higher-order chromatin and nuclear architecture, and to integrate all of these features into a systems-based model of the disease. On the one hand, MODHEP's experimental plan requires expertise in genomics, epigenomics and nuclear architecture, and on the other hand, expertise in cancer biology and animal models. MODHEP is a very important research project, both in its novel approach to understanding cancer, but also because liver cancer, of which HCC is the most common, is a big global killer. There are 750,000 cases of the disease every year in the world, making it the 7th most common cancer, while death follows in 600,000 of those cases, making it the 3rd leading cancer-related cause of death worldwide. Like any other cancer, it develops when there is a mutation to the cellular mechanism that causes the cell to replicate at a higher rate and can result in the cell avoiding apoptosis, the natural or programmed death of a cell. In all cases, early detection will improve chances of successful treatment. New drug treatments that target specific parts of cancer cells are also helping to improve survival rates. But early detection and better treatment both depend on our understanding of how the disease forms at the molecular level. And this is exactly what MODHEP is all about, and how its important work helps in the fight against cancer. The project is studying HCC by looking at cancer at its very basic level, in the cells, molecules and genes that make up our bodies. In terms of liver cancer, there have already been many studies into gene expression profiling. Gene expression profiling is the measurement of the activity, the expression, of thousands of genes at once to create a global picture of cellular function. These profiles can, for example, distinguish between cells that are actively dividing or show how the cells react to a particular treatment. Studies have already identified several molecular pathways that are deregulated and/or mutated in HCC. In other words, certain actions among molecules in a cell have been seen to be specific to HCC. But what actually causes these molecules to behave this way in the disease remains largely unknown. At the molecular level, tumours develop because of changes in sequence, structure, organisation, accessibility and ultimately usage of the genome. So MODHEP is looking to develop models to understand why genomes become abnormally programmed and how this controls the pathological behaviour of cancer cells. To do this, MODHEP has been working on an analysis of the nuclear changes occurring in 2 complementary multi-stage models of liver cancer in mice. As well as this, human samples of both healthy and cancerous liver tissue have been studied in order to look at differences in gene expression between the 2. Together, these studies are providing a complete description of the changes that occur at both the pre-tumour and the tumour stage of the disease. The data from these studies has been brought together using innovative systems biology-based approaches. Systems biology focuses on complex interactions within biological systems. The aim has been to link all this experimental data with the molecular pathways of HCC. Once this is done, the project will be able to assess the validity of the main features of this model network in the human disease.